Let's get started today. Welcome, everybody. Thank you for joining. This is the April 17th meeting of the IMAG MSM GLIMPRINT series of mini seminars, something like our 153rd one of these. So thank you for the people who've been here from the beginning. As always, I need to remind people that the meeting is live streamed and recorded on YouTube and available in an edited form afterwards, so please be aware of that. Tom Helikar and myself, Jim Sluka and Lorenzo are always available to take your suggestions, comments, critiques, ideas. Please don't hesitate to suggest ways we can make things more effective and more useful for you. If you have trouble reaching us, you can find us on the websites. We're always eager to hear from you. We have a variety of communication channels. They probably work better if people use them. There is the GLIMPRINT homepage, which Jim Sluka has worked on a lot. There is the old IMAG wiki page. There's a LinkedIn page. There's the YouTube channel. Please help us get the information out there. Especially if you gave a talk, please feel free to circulate the videos of the talk. Let people know about it. Something we've been fantasizing about for several years was having a logo. Tom has been working with ChatGPT and others on trying to develop a logo. We have some provisional logo ideas. If somebody would like to comment on those, by all means, let us know. You'll see we now have a little temporary logo at the bottom left of our slides. That's so doesn't seem like much, but our European colleagues are very good at generating logos, so we should do the same. If you have a publication to share that's related to digital twins, multiscale modeling, immune system, could be experimental, clinical, otherwise, please send us on to us, especially if it grew out of efforts through the working group. We could put it up on the IMAG site and on the GLIMPRINT site and help publicize that. Send it to Jim Sluka, jsluka@iu.edu, and we'll do our best to get the word out for you. Here's the GLIMPRINT site. If you have seminars, meeting announcements, workshops or classes, job opportunities, funding announcements, we can also put them up. So do please help us populate that site and generate a little bit of interest in that site. The other thing that I have to keep reminding people of, it's going to be a shock after 5 years, 4.5 years, is going to be that to accommodate our European colleagues, and I'm afraid disaccommodate people on the West Coast, we are going to be, at least for the time being, moving the seminar time from 3 p.m. Thursdays to 10 a.m. Thursdays. We did a survey, and the people who responded said that that worked for them. I hope it will be all right for the majority of people. But this will allow us to coordinate with Anna Nirakis's efforts in RDA on immune digital twins and be able to co-host seminars. Otherwise, the timing is 9 p.m. for our European colleagues is difficult. I realize it's not so convenient for our California colleagues. And that new time is supposed to start May 15th. A few other announcements. I always have my annual user training workshop and hackathon on CompuCell. That's going to be from July 28th to August 10th this year. If you have people, students, others who would like to learn how to use CompuCell modeling software, we would be very grateful to have you send them our way. We have the Open VT Reproducible Agent-Based Multicellular Modeling Competition. That is still available for people to submit to. If you have been doing multicellular modeling on your own or using any framework, it could be NetLogo, it could be PhysiCell, it could be Biocellian, it could be our friends at Moffitt's HAL, for example, anything that has multiple cells and agents in it, and that you've published since 2023, and where you've made the code available publicly. We would love to have that in competition. We're going to have people who win that speak here in this seminar series. There are going to be small cash prizes, and there's going to be a formal announcement at the SMB meeting this summer. There's also the Biomodels Model of the Year competition led by Sharif, and that also has an extended deadline. The applications for both of these are not onerous. They're similar. They're not exactly the same, but they're pretty close. Most things that would qualify for our open VT modeling competition would also qualify for the biomodels competition. The biomodels tends to be heavier on network models rather than agent-based models, but please do take a look at both of them. We have some great applications already, but we'd always love to see more from the community. At the SMB meeting, we're going to have a satellite workshop on model sharing. There also are going to be 3 mini symposia sponsored by GLIMPRINT covering immune digital twins. And so we hope to see you at both of those events. There is a recent call by Grace Peng at NIH that there is going to be a special session coming up on cyber infrastructure and emerging technologies in biomedical research and ISMB. And if people are interested in that, they should get in touch with the appropriate people. There's certainly some overlap of interests there, and she asked us to let people know about that. The FDA has an initiative on novel approach methodologies, new approach methodologies in drug development and toxicology. That, again, is of interest to people in this community, and people should take a look at those FDA initiatives and see if they're appropriate for what you're doing. Moffitt is hiring, and again, there are many people here. I see some people who are either at Moffitt or were at Moffitt in the room. So perhaps people could talk to them about the details. But there are some openings at Moffitt. Very fine place to do multicellular, multiscale modeling related to cancer. And so people should contact them if you're interested. Coming up, our next meeting on May 1st will be Marcus Covert, of course, very famous for his work in the E. coli whole cell modeling project. That's just gotten a big expansion, and they're going to be bootstrapping that. We have some other interesting topics coming up. We still don't have a speaker for May 15th. As always, you hear me say this every 2 weeks. Please suggest yourselves, your students, your postdocs. If you've spoken before and you want to give us an update or talk about something you haven't spoken before, we're happy to hear it again. Also, if there are people you'd like to hear from in industry, experimentalists, clinicians, or modelers, please let us know. Send us the names and contact information. Lorenzo is very effective at getting people to come to speak. We want to make sure that the people who come and speak in this forum are people that you want to hear. And we realize it's a very diverse community, and so the more suggestions we have from our audience, the more we can meet the needs of the community. We will have a steering committee meeting tomorrow at 11. As usual, I'm reminding you that starting May 15th, again, we're moving the seminar earlier in the day. Are there any other announcements before we go on to our seminar today? Again, I want to thank everybody for coming today. I really appreciate it. We've have back-to-back talks on radiation oncology, which is unusual for us to have two talks sequentially on the same general area. And so I appreciate people coming today. And so I don't want to take any more time from our speaker today. We're going to be hearing about multiscale digital twins for personalized radiation therapy. And I'll turn the screen share over to you.
Hello, everyone. So my name is Ruirui Liu. I'm a assistant professor in University of Nebraska Medical Center. So today I'd like to share some of my thoughts on multiscale digital twins for personalized radiation therapy. So I would briefly talk about digital twin for healthcare. I mean, probably most of the audience today know this, so I will just very briefly talk about this, and then I will just majorly talk about the um, two items. The first one is the multiscale digital twin applications in radiation oncology or radiation therapy, and then I will spend a little bit of time in AI-powered digital twin technology. Let me go to the first one. So what is a digital twin? A digital twin is a dynamic virtual representation of a physical entity created using real-time data and sophisticated computational models. And for radiation therapy, and one of the particularly important um, like a technology nowadays is called patient-specific digital twins, because it can integrate like a patient physiology and sometimes immunology information to patient, and we can use this data to run some computational simulations, and we can test some maybe just like a proposed design, proposed radiation treatment protocol or some new type of treatment modalities. And the reason why it's powerful that with the computational power increase and also a lot of detail, data collected in clinical settings, and nowadays we'll be able to, generate very fine patient-specific models, and we can use this to customize treatment. And in one of the like a new archive paper, and in this review paper, it's give a very nice like conceptual layout. For example, like digital concept says there's one side is called a real world. So basically, it can include the patient anatomy, patient physiology, and also can include some treatment modalities from different treatment machines, and even a general like version of a hospital. And correspondingly, there will be a like virtual copies of all these items, and there will be a virtual human body, with all the details from like physiology information and anatomy information. You also can have a virtual version of machines and virtual version of hospitals. So this is basically it's like mirroring the real world into computational simulation models. And with this right side map, it shows there's many uh, digital models and tailored for specific maybe just reasons. For example, there's some digital twins for kidneys, some digital twins for prostate, livers, and intestine, etc. So basically, there are some many um, like digital twins for different parts of a patient nowadays over there. And a few like a research trajectory like related to digital twin in radiation therapy. So what I found is that most original version or like similar concept as digital twins is called in silico medicine. And there's a very, I think it's almost one, almost about 4 years ago, there's a first paper published, and the paper is called Modeling Tumor Growth and Irradiation Response in Virtual and using high performance computing and web-based technology. So in this paper, this author proposed a term called in silico medicine, and later on that evolved into in silico oncology. So I think that's one of the research trajectory is similar as digital twin in radiation oncology nowadays. Another one is we know that's called computational oncology, and this one I think is a very active research nowadays. And in the United States, there are a few big groups. For example, from University of Texas Austin and University of Chicago, and also very famous cancer center, Memorial Sloan Kettering. And all of those centers have very good computational oncology program. So primarily, they develop mathematical models or computational models to simulate certain type of radiation therapy treatment. There's a similar term also called mathematical oncology. I would say it's pretty much similar as computational oncology. Maybe a mathematical oncology is more, I would say, it's more theoretical investigations. It's more, I mean, style is more mathematical theory development. And in the United States, there's one center called Moffitt, and they have very good in mathematical oncology studies. Another research trajectory is called computational radiation biology, and there's a few centers, but there's still, I don't think there's like well-known centers nowadays for this like computational radiation biology. And personally, I delved into this field since I was a PhD student back at Oregon State University, and at that time, we pretty much using the computation models to study the microdosimetry effect and apply this kind of microdosimetry or even nanodosimetry concept into radiation therapy. I see this is also one of the research trajectory, similar to digital twin in radiation oncology now. And I also want to spend some time talk about this, like enthusiasm from industry. So this digital twin in healthcare or like digital twin in radiation therapy, I would say it will be boosted or is still in the process of boosting by this kind of enthusiasm from industry. So there's very ambitious plans nowadays trying to using the advanced simulation models or platform to study the medicine related problem. For example, last year the Nobel Prize went to the AlphaFold members, those the people developing very advanced deep learning based models in order to predict the proteins, and this can be very, very powerful to, speed up the drug development, just for cancer treatment. And also, Facebook also has plans, for example, building a, digital version of human, and trying to use a digital version of human to eradicate some, human disease. And of course, NVIDIA as one of the superpower in computational structure nowadays, they also have very ambitious plan to build human digital twin models. Of course, there are so many emerging funding opportunities nowadays, for example, from NIH and also National Science Foundations. Yeah, then let me just go to my first item today is multi-scale digital twin applications in radiation oncology. So status of multi-scale modeling. So understanding and optimizing clinical radiation therapy is very important for modern oncological treatment. And the digital twin emerged as a transformative tool for radiation biology and also radiation therapy. It's essential to develop multi-scale models that can capture subcellular, cellular, and tissue level response to radiation. So the key is that why we call it multi-scale modeling is that so basically the key is that in order to simulate, for example, radiation response for tumors, and you need to consider this different level of concept for example, you may consider the DNA damage that might be from subcellular scale. And for example, the cellular response, where a cell will be killed or a cell will be still surviving, there will be a cellular level questions. And for like if you study the whole tumor or treatment, effectiveness, that is in tissue level problem. So basically, we need to study all the problem in these levels, and most likely we need to combine all of them into a simple, like a single platform or structure. So this requires a high performance computation platform that can, help researchers in the clinician to create, explore, and validate these models, and then further apply these, models into the clinical settings. And there's already some like I would say semi mature platforms, but all these models or platform has its own limitations. At the subcellular level, there's a well known like Monte Carlo structure code, for example, like gene for DNA and retracts and pertracts, and some other you know platforms. But for this platform, some of them are open source, some of them are not open source, and also this kind of structure code, you need to just develop your own extensions based on this platform in order to tailor the development to your own problem. So basically, a lot of code published by the previous users or like developers maybe is not ready for you to use directly. And at the multicellular level, there's some agent-based modelings. For example, as Dr. Glayer named initially, is for example, physical cell models and also computer cell models. But both of them are lacking radiation transport simulations. So nowadays, if you want to apply this multicellular platform and use them for your radiation therapy modelings, you need to develop your own radiation transport simulations and then couple with them. And so our work in developing this computational platform actually can be traced back to 2017. And at that time, I was still working as a PhD student at Oregon State University. So I actually just that time I visited Dr. Glader's lab about like 4 months into 2015, and in his lab, so I primarily worked to develop one of the bridging softwares. We call that Red Cell, and Red Cell actually is first computational platform that integrating the gene for and the gene for DNA. All like gene for and the gene for DNA is a general radiation transport platform, which we can use them to do the dose calculation and also the radiation-induced DNA damage simulations. And so basically, our idea is combine this gene for and the gene for DNA platform to CC3D in order to achieve the multi-scale radiation biology and radiation therapy models. So the red cell actually can achieve the like 3 level of modeling. So for subcellular modeling, so it can just simulate the organelle, for example, nucleus and cytoplasma or like mitochondria. Basically, it's enable you to create and add these cell organelles into a single cell or multi cells, and also we can use that to calculate the DNA damage introduced by the radiation, and also can simulate the kinetic DNA repair. For the multicellular scale modeling, so that time we developed a population level radiation dose and DNA damage analysis and models, and we use that model we can predict the cells phenotype transitions. I will just introduce the detail a little bit later. And in the tissue level modelings, so basically that at this level we couple with the CC3D in order to predict the tumor response. So then I would just talk about a few applications for all these three levels for the subcellular and multicellular simulations. So the red cell actually can just simulate a single cell model with very detailed complete human genome, like a spatial structure. So basically, you can build a very accurate DNA spatial structures and then apply the radiation transport to the spatial structures, and I can calculate, for example, the what's the double strand break yield and what's the single strand break yield, and we also can just, predict, what's the contributions from the direct radiation induced DNA damage, and how much from the indirect DNA damage by the free radicals. And so, for example, this figure shows, for example, the like a figure A shows a single cell models. Here we add the cytoplasm and also the cell nucleus, and also we can initiate radiation transport within this cell. Of course, we also can just simulate multi-cell cells. For example, the figure C shows, I think this is close to 2,000 cells, which is conformed together as like a secured. We also can do radiation transport for this many number of cells, and we can just get the dose for each cell, and also can get the DNA radiation induced DNA damage for each cell. For example, we can know the double-stranded break yield for each of the cells, and use this DNA damage information to predict how the cell will be after radiation transport. For multicellular response after radiation, red cell actually can just simulate how cell will be after radiation transport, and it's relying on a few steps of simulations. For example, in the initial version of red cell, we don't have the P15, but in the newer version of red cell we are working on, we add P15 cell response, and so this is the newer development, and we add the microenvironment simulations, and the new version will also add modified cellular phenotype transition models. So for the P50 simulations, so basically we just use the kinetic simulations for this kind of P50 response structures, and I think this is well studied. So basically we just work on this and just apply this to a multicellular modeling. For the tumor and tissue microenvironment, so we now it's like primarily just solving a reaction diffusion process. So basically bounceable coupled PDEs, and we just working now is using the CC3D's engine to solve this. Then I would just spend a little bit more time on this newer version model called cellular phenotype transitions. So a cellular response to radiation can be in different types. For example, cell cycle rest or like cell apoptosis. And usually this can be modeled with the explicit P15 protein networks. But this approach is very computationally expensive for multicellular system at a clinical scale. So nowadays there's a kind of I think agreed in our field that if you want to consider your model is clinical scales, so there must be over 10 to 9 cells. So if less than that, it's hard to justify your models is clinical scales. So the issue or challenge now, for example, if you want to apply radiation transport, DNA damage, and also P15 protein network for this large number of cells, it's very computationally expensive. So the new design in the red cell now is we incorporate a probability-based cell-state model to predict the post-radiation response without simulating detailed DNA repair or the P50 networks. So the key idea I will elaborate a little bit. So basically, we classify the three possible radiation-induced cell phenotypes. For example, of course, if there's no damage or damage is minimal, probably the cell still will be healthy or alive. Cell cycle also can be halted or perturbed, and we call this as arrested. But if the DNA damage is repairable, so the cell will be dead or killed. So could be the apoptosis or necrosis. So basically, we classify these different possible cell phenotypes, and then we group them into three possible states. One is called healthy state, another one called arrested state, and then third one is dead state. So then we introduce the cell state transition like a route. So we can just have healthy cell directly jump to arrested or dead, and the arrested cell also can be repaired to a healthy state or also can jump to dead state, and that remains terminal. And in order to just quantify this probability like transition probabilities, we introduce a cell state energy. And the cell state energy is combining the direct energy and also indirect energy. Direct energy is corresponding to the radiation directly induced DNA damage, and indirect energy is corresponding to the, for example, the indirect effect, such as the free radical induced DNA damage. And so there's a few equations in order to just calculate this. But the key idea that so basically you use this kind of mathematical formula to predict this kind of transition diagrams. So for example, healthy state can go to arrested state, also can go to dead state, and arrested can go to healthy state. So this model produces biological plausible result compared to experimental data, and its parameters are simplistically estimated. But the uncertainty analysis is lacking. This is the early version of the red cell development. So these days we refine this model to enhance the accuracy and efficiency in simulating the phenotype of transitions. How we do that is we use the P15 protein network simulation to simulate a smaller scale of cells. For example, just 10 to third, maybe like 1,000 or 10,000 cells. So it's a smaller scale, and so using this smaller scale system, we just use explicitly modeling. For example, just directly calculating the radiation-induced DNA damage, also incorporating the DNA damage repair, and also the explicit P15 protein networks. So with this, we actually can just get kind of a more accurate or like more close to the experimental observation result. And we use this, maybe we can call it intermediate, like a number of cells, and we can just use this result to just feed the some of the very important parameters into that statistical models. So for example, we can just run these smaller scale cells from 1,000 or 10,000, and we, as I mentioned, just you go through all these kind of very explicit simulations, and then you can just group the potential phenotypes with all the state energies. So basically, you can just using a parameter fitting to get kind of more accurate parameters for the models. And then with this, you can just apply to a very large number of multi-scale cell system. For example, go to 10 to 19 cells, because you don't need to spend a whole lot of time calculating the radiation damage and also the P15 responses now. So basically, you just apply that probability transition models for phenotype transitions for each cell. Now it's basically very direct calculations. So with this, we actually can just simulate the multicellular response after radiation. So this is still a list of few like simulations. So from the left figure here shows the a multicellular system. I think is about 10,000 cells, and we just use electrons to irradiate this multicellular system, and then we try to predict how the cellular phase distributions will be. So, for example, we this different color curve corresponding to the different phases, and the axis is the time. So basically, you can just, or we can call this like cell phase spectrum with time. So you can see like but the figure is a little bit busy. But the key idea that with this simulation platform nowadays, you actually can simulate the cellular response, and the cellular response could be different kind of endpoint you want to observe or predict. So here we want to see the cell phase spectrum. So the key idea you can see here is with increasing the dose to the cell, you can see the cell phase spectrum is severely perturbed. It's no longer as, for example, the A here shows this kind of very rhythmic, pattern. But with the radiation introduced, the system is perturbed. So some cell may be killed, or some cell is the transition is like um, perturbed. So the phase is different. And the right figure here shows the cell state spectrum. So for example, A here is the spectrum without any dose, and all others is the cell state spectrum, or we call it the phenotype spectrum, with different level of radiation dose. So you can see, for example, the red curve is the dead cell. So you can see with the radiation dose growing, the dead cell is pretty much like dominant. So basically, more and more cell are killed. And most importantly, we can see, so in radiation therapy or radiation biology, one of the very common important, analysis is called cell survival fraction curve. So basically, you seed certain number of cell into a petri dish or even 3D cell culture space, and then just grow the cell. It's very healthy, and then at a certain point, you just deliver radiation to the cell system, with given certain level of dose, and then you just observe, the cell survival fractions for this cell system, and usually you plot the curve, with respect to the dose you're given to the system, and also the cell survival fractions. So generally, it will give you a exponential shape of cell survival fractions. So we just use our system to simulate this, and you can see we can get pretty reasonable cell survival fraction curve. But most importantly, for our system, we can just apply this to very interesting radiation induced effect. For example, we apply this to study the radiation induced bystander effect. And the red curve is the cell survival fraction curve without considering the radiation-induced bystander effect, and the blue is the cell survival fraction curve with considering the radiation-induced bystander effect. So most striking point is that at lower those regions there will be a dip. So basically, the more fraction of cell will be killed if you consider the radiation-induced bystander effect, and that's actually is like overlapping the experimental observations. So that's pretty much that's just like something like, for example, with radiation-induced bystander effect, there will be like higher sensitivities in lower dose regions. And the key from this study that our platform actually can replicate some of the well-known phenomena or effects in the radiation biology study and also radiation therapy study. So the coupled simulation toolkit, as I mentioned, the red cell. So if we just break the whole system, you can see there's a few elements there. So it has cell biology simulation engine and has cellular tissue microenvironment simulation engine, radiation transport engine, and also cellular radiation response engine. So this has all these engines combined together. We can just simulate or we can build a multicellular digital digital models for radiation biology and radiation therapy. And particularly for cell biology simulation engines, we couple computational 3D models. And we all also working on coupling with tissue force and also another platform developed in Dr. Glader's lab. And we are also working on some GPU accelerated DNA damage simulations. So this is some about the platform. And here I'd like to just emphasize one of our previous published studies. This published, I think, almost 3 years ago, using the red cell coupling with cell CC3D. So that time we just use CC3D and also red cell to study a vascular tumor response after radiations. So the reason why we have this is that at that time, I think this is one of the nicest published model from CC3D. Basically, using CC3D to simulate a vascular tumors, you can see very interesting. It's more like kind of a micro level of the tumor, tumor rendering in the 3D space. So the idea that how about we apply this to a radiation therapy settings. So basically we just deliver like a photon dose using X-rays, and basically irradiate this small piece of vascular tumor. And this vascular tumor at that time, the CC3D has I think 4 or 5 cellular types. So what we try to see that how about we deliver different level dose to the 3D vascular models and see how this three-dimensional vascular tumor response. And we can just get this tumor load spectrum. So basically you can see a different level of dose corresponding to different tumor load spectrum. And you also can see, the three-dimensional rendering shape. So basically, that's very interesting. You see like with this 15 gray, and this is with 20 gray, this with 25 gray, and this one is without any dose. So without any dose, so basically, if you go for a certain number of many color steps or certain length time, the CIS3D will give you this shape of this vascular tumor. But you can see with this different level dose, the three-dimensional rendering is no longer same as the original one. And further, we apply this to testing some like just like a hypothetical radiation treatment protocols. For example, we say how about deliver 50 gray in 5 fractions, and also 40 gray in 2 fractions. So we can plot this kind of tumor load. We also can see how the three-dimensional rendering look like. Definitely it will be different. So for example, the blue is 40 gray in 5 fractions. So basically same dose but delivering 5 times. And you also can deliver same dose, but in two times. And you can see with these 5 fractions, maybe it's killing the tumor cells more effective. And this is this is still old version of CIS3D, and coupling with the Computational 3D, you can see after the system going on, and we can see this vascular tumor is growing, and you can see the tumor load, but here is at a certain time step with start to irradiate this cell, you see that radiation track structures, and you can see with one dose giving the tumor load it will be reduced. So this is basically is a simulation video, and we also apply that platform to a that time also a good CC3D applications called like eggs gastrulation simulations. So we just applied this simulation platform to this. Oh, sorry. We also can see, this is the CC3D simulating the egg gastrulation. So this is the the time going on, there will be uh, a tip, is emerging in the center of this egg. But with at a certain step, we introduce this radiation transport to system. You can see the some of the cell is killed, and we render that into a black dot. And you will see there will be some like difficulty for the cell forming that gastrulation, this tip. Yeah, this is like two of the like applications we've done before, and one of them is published already, and one of them we put into archive now. So with this going on, so I will share a little bit of thoughts about, for example, how to build a digital twin for radiation therapy. So as I mentioned, a digital for radiation therapy, you have to consider these different spatial levels. So, for example, at the very maximum level could be a human's body, and in order to study some certain disease, you can go to go to certain organs. For example, you can just study vascular tumor tissues, and in the multicellular scale, you can use this to simulate the multicellular DNA damage and radiation transport. Of course, if you go to like even further, you can just study the DNA damage. So basically, you need to consider these different spatial levels, but the very challenging is that how you're gonna efficiently combine all of them together into a single platform. So this will be very challenging. And also, how can we apply this digital twin for the radiation therapies? So I propose that in order to successfully apply digital twin into a radiation therapy, you need to at least include 5 key elements. So first one, you need to have models to give you very accurate spatial or geometrical information, such as your anatomy, or for example, in radiation therapy, like some contours of your target. And you also need to consider temporal data. For example, because with different time step, patients' organs or tumors will change. Just imagine, for example, a lung tumor, patients with patients' brace, the tumor actually can move, right? So this is kind of a temporal pattern. So you need to have good models to consider this. And you also need to consider the material properties, because for radiation therapy, you need to get very accurate dose. And those calculations is very largely are very importantly dependent on the material properties. If you don't have a accurate material properties, you cannot get very accurate dose calculations. You also need to consider functionality within the physical environment. So for example, just like some type of radiation therapy, for example, a brachytherapy, you need to insert some needles to the organs or some like lesion site, and that can lead to some functionality change. For example, some of the some of the organs may be damaged if you are not consider some potential applications. So in order to consider this, you also need to have models consider the functionality. And of course, the last elements, I call that interactions with other physical object. This can be there's good examples still, for example, the brachytherapy for the prostate like a high dose rate therapy. Usually, for example, you need to insert a ultrasound probe from the rectal region, and after you can imagine after that probe inserted, the process shape will be different as original shape. So this is a physical interactions happening. So basically, in order to accurately consider this, you also need to consider some interactions. And then I will just start to just illustrate how we consider these different five elements using this example. I call that a discipline for prostate ultrasound based high dose rate brachytherapy. So for example, as I mentioned, the first one will be having very accurate spatial geometry models. So for example, this is some of our studies. So we developed an onboard patient model. So basically, this is like a patient like a prostate phantom. I mean, so for example, we can just generate these three-dimensional like patient models using the ultrasound images. So basically, and also from images you can see the different insert needles. So with this model, you will be getting very good spatial and geometry information for your treatment. And one example is temporal model. So for example, with the needle insertion, as I mentioned, the patient's anatomy will be changed. So for example, the prostate will be changed, right? So we developed a dynamic deformer registration from ultrasound to patient MRI. So basically, we can just get this like in time, contours. If we consider all the like anatomy change. Of course, the material and function models, materials. So for this type of study, we treat the patient as just patient material as water. But this is just a clinically defined nowadays within the world. So basically, we just for this types treatment, we use water to represent the patient materials. But in other type radiation therapy treatment, you cannot just use water to represent the body. You need to consider very fine, material types. What I want to show here is that definitely for this type study, we just use water. That's enough. And for the function models, for example, as I mentioned, different kind of like normal organs and their effect and their response after radiation. So you need to consider this. So basically, you need to get the all the kind of normal organs contour in order to consider the implications of this type of radiation therapy. And the interaction model, for example, here I give example of this published data, and they use mathematical models to study, for example, after the needle inserted to the prostate, how the prostate shape will be changed. So basically, this is a good example for showing you need to have interaction models. And here I spent a little bit word for emphasizing the effect or the good applications for digital in radiation therapy, that we can run virtual clinical trial using digital. So the benefit can be different. So for example, the most obvious one, you can save time and the cost, because if we develop the personalized digital and we design that well, for consider that the five key elements, and we can use that as a virtual patient, pretty much like have good spatial geometry information, physiological information, physical interaction, and so on. And we can use that to test different type radiation therapy protocols or like treatment modalities. So first you can save time and also cost. And also very, very important, I would argue that we can increase the efficiency of developing new treatment methodology. Sorry, there's typo. So basically, you don't need to consider, for example, like the the blockings from IRB or something, get approved from FDA. But we just run this from a computer version of simulations. This can just you know sufficiently increase your testing efficiency. And lastly, I want to spend a little bit time in this AI-powered digital technology. So, what AI can help us here? So, here I also trying to correlating this with that 5 key elements I proposed. So, first, at body scale, and AI can aid identifying suitable patient and making diagnosis, treatment planning, and etc. So this is happening nowadays. So it's no longer new, but it's with the very powerful models emerging and the computational power increase, and this can be becoming ever nicer and nicer. And the tissue scale, so the tissue response is related to solving complex, PDEs, as I mentioned, for example, the reaction diffusion models, and AI can just solve this. So that's one of the direct applications, but there will be more. Here I just list one of the potential contributions from AI. And the 5 elements I proposed, most of them are relying on AI, as I just, for example, just showed you that process brachy therapy case. And a lot of models we developing nowadays is relying on deep learning. And multicellular scale, validating the multicellular simulation models relates to comparing the simulation result and experiment data from cellular radiation, for example, microbeam radiation, etc. And the AI can aid us conducting results analysis and data validations. And subcellular scale, there's some AI models that actually can very fastly predict radiation induced DNA damage and more. So that's why AI can help us a lot in order to apply this digital technology in radiation therapy. And from this archive paper, and I want to follow this figure and share with you guys, is that with this time going on, and there's more and more these kind of digital publications. And if you dig to this paper, you will see like all these kind of publications, pretty much all of them are AI-based models, even either deep learning or some sort of artificial intelligence models. So that's also another strong argument that AI is helping digital models in healthcare or radiation oncology. And also, yeah, this table shows, for example, this is different, clinical sites or like lesion sites, these trends, and also you can see the computational models. You can see a lot of AI models, UNet, some other type of artificial intelligence models. And there's a new paper in this study actually directly using the deep learning models to calculate the radiation-induced DNA damage. Yeah, so here's a few like conclusion points, and I would say undoubtedly the development of these digital twins will better promote the advancement of radiation therapy, and these twins can help us understand, observe, and even predict patient response, which can enable patient-specific treatment or, as I mentioned, the clinical trials. Building the framework for digital twin is a very complicated process, and it's related to many fields, and it needs a cross-platform integration. For example, our development is a good example, like integrating the radiation transport and also the tissue models from CC3D. And of course, you cannot forget or neglect artificial intelligence. And this is very powerful nowadays, and it can provide a tremendous benefit for us building a digital framework. Yeah, from here I will stop here. If you have any questions, I will be happy to answer. Thank you.
Thank you very much. The floor is open for questions.
When was your recent application of your model to like a clinical setting?
Oh, sorry. So your question is what's reason applying models in clinical settings, right?
I said, what's the recent observation have you seen in your model in a clinical setting? Like,
Oh, so I actually don't know, for example, somebody applied, for example, my models into clinical settings. Yeah, there's some citations about, for example, red cell. But most of them, it's not in, for example, clinical settings yet.
So I think you talked about it a bit, but maybe it's worth coming back to that a little bit more, which is what do you think the key needs are to get this technology from where it is now as a research tool to something where you could deploy it clinically?
Yeah, that's a very good question. So I actually has been like since I came to University of Nebraska Medical Center, I've been like interacting with, for example, lab radiation biologists and also the radiation oncologists. So for example, I talk about this with them, and they know this concept, and but most of them I think still have like concerns, for example, like how to validate these models in, for example, in order to directly apply to clinical settings. Really, the wet lab radiation biologist, I think it's more easy to, I think, accept our ideas because that is like cell simulations. For example, we can simulate the cell survival fraction curve or like cell cycle arrest. Or some sort of cellular effect. That's a kind of a, for them it's easy to accept our proposals. For radiation oncologists, I think they seems like have concerns. It's just like different levels, like for example, primarily our so far studies is still in either in a very tiny scale. It's like a 10 sound or like a few sound cells, or like maybe 100 sound cells. Is a small tiny piece of tissue. And the reality is that in real, for example, clinical settings, like treating a tumor, they don't consider that like microdosimetry or like that small level of concept anymore. It is like patient come in and get the images like CT, MR, or ultrasound, and there's like a well-defined protocol they directly use. There are questions that what you actually can help them. That is something I cannot directly answer them because the clinical settings, most of them is based on the like long years of clinical knowledge accumulations. Yeah, definitely they know cells fraction curve, different cells sensitivity in terms of radiation, but in the clinical setting they don't think about that that into that detail. So basically still giving impression that into two separate world. We are very enthusiastic about the functionality or effectiveness of this cell multiscale modelings, but we are not that fancy or like effective yet to convince them we can directly help them. That's my impression now.
So it would still take some effort to be able to make this clinically actionable.
Yes. Yes.
What about including additional mechanisms? I mean, this community talks a lot about immune modeling, for example. So, we're thinking
Yeah.
you talked a little bit about bystander effect, but you might want to think of what about combination therapies where you combine radiation with chemotherapeutics or with immunotherapies, for example. Would those
Yeah.
be cases where this kind of modeling might be more useful? Or well, useful may be the wrong word, but might have more obvious value to a clinician.
Yes, that's very good observation. Definitely. So I would say from the modeling realm, I think that's a pretty ready now. For example, we couple either, for example, chemotherapy, immunotherapy, and radiation therapy, and we can study the synergetic effectiveness or like try or test this kind of combined protocols for certain type of disease. That's for sure. It's working. One of the I think is most promising or ready for us to apply that is I can think of is carbon therapy. And in United States, I think I heard in Mayo Clinic in Florida, they will be having the first carbon therapy facilities. And for carbon therapy, one of their like key issues that you need to get the effective biological effective factors for different type of like particles, or for example like carbon, photon, electrons. But for carbons, you definitely need to have these biological effective factors in order to make a treatment plan. The things are very nice that nowadays, if you look around the whole world, for example, the most centers having carbon therapies, most of them still using mathematical models to calculate that biological effective factors, or we call it RBE factors for carbon. And that gave me impression that, for example, our multicellular model or multiscale models actually can be directly applied to that settings. So I can foresee that carbon therapy or like neutron therapy is like two of the direction we can find more application opportunities.
Thank you. I remember years ago hearing some work at Vanderbilt, Vito Quaranta and the people around him thinking about trying to model heavy nuclear radiation as cancer therapy. I guess, I mean, proton radiation therapy is fairly common. I think heavy nuclei are not so common yet.
Yes. So that's why I think there's a good opportunity for us to just develop models for carbon therapy. And also for carbon therapy, there's one of their basic mechanism why carbon therapy is better or like more effective for certain disease, that carbon therapy can initiate some type of immune induce the immune process within the body. So to for example align with your argument, we actually can just develop models, incorporating immune response, radiation response, and also some type of response, and that is very good models just tailored for carbon therapies.
Okay, well then I want to thank our speaker for today. I really appreciate you coming to give us a talk. Hope we'll maybe have you back in the future and have you speak again and tell us how your work is developing. And I want to thank everyone for coming today, and we'll see you in 2 weeks.